Hi, I'm Cade Brenneman, and this is my mitosis project. The cell life cycle, as you can see, it starts with interphase, and there are three main phases of interphase. Interphase then enters mitosis, which has four main stages, and then cytokinesis is the final factor. Um, Interphase, like I said, is the first stage of the cell cycle. It's where most of the cellular activities occur. It has three stages. The first stage is the G1 phase. This is where the cell grows in mass, and that means the cytoplasmic components double. The second stage is the S stage. This is where DNA replication occurs. The chromosome number duplicates. The last stage is the G2 stage. This is the interval immediately after DNA replication. The cell prepares for division and is prepared to enter mitosis. Mitosis is the process after interphase. Mitosis can be described as the cell division where one cell divides to produce two new cells. It maintains the chromosome number. There are four unique stages of mitosis: prophase, metaphase, anaphase, and telophase. The first phase is prophase. This is where duplicated chromosomes become thread-like as they start to condense. Microtubules form a bipolar spindle, and the nuclear envelope starts to break apart. After prophase, there is a short phase known as the transition to metaphase. This is where microtubules form one spindle pole, and they harness to a chromatid of both the chromosomes. So they extend from both poles and they grow and overlap at the spindle's midpoint. So metaphase is the next stage. This is where all the chromosomes become aligned midway between the two spindle poles. Chromosomes in this stage are in the most condensed form. After metaphase is anaphase. This is where sister chromatids detach from each other. The spindles move them toward opposite poles, and the microtubules that overlap at the spindle's midpoint slide past each other. So they push poles farther apart, and the motor proteins also drive movements. So the phase after anaphase is telophase. This is where two identical clusters, one chromosome from each type, reach opposite spindle poles. The nuclear envelope forms around each cluster, and both of the new nuclei have the parental chromosome number. So after telophase happens, a process called cytokinesis occurs. Many people think this is considered a part of mitosis, but it's not. It just deals with the division of nuclear DNA material. It is the last step of the cell cycle, and it is known for dividing the cytoplasm. Meiosis and sexual reproduction. Meiosis is the nuclear division mechanism that precedes gamete formation in eukaryotic cells, which it has the parental chromosome number. Meiosis occurs in sex cells mainly to maintain the haploid nature of gametes. Sexual reproducers inherit pairs of chromosomes, one from the maternal parent and one from the paternal parent. These pairs are homologous, which means the same. The female has the sex chromosomes XX, and the male has the sex chromosomes XY. So, a little tour of meiosis is there's two divisions, meiosis 1 and meiosis 2, which divides the parental chromosome number by 2. Each forthcoming gamete that is formed is haploid. So, meiosis 1, it is the first nuclear division. It has four stages just like mitosis: prophase, metaphase, and anaphase, and telophase. So, the first stage, prophase 1. This is where chromosomes condense and align tightly with their homologies. Each homologous pair undergoes crossing over. Crossing over is a big process. This is basically where two chromosomes of a homologous pair exchange equal segments with each other. Um, Crossing over is really important because when chromosomes are replicated into two strands, they're called sister chromatids. So, and then in prophase 1, microtubules form the bipolar spindle, and one pair of the centrioles moves to the other side of the nucleus. So, the next phase is metaphase 1. In metaphase 1, microtubules form both pole position all pairs of homologous chromosomes at the spindle equator. So, they line up and around the equator midway between the two poles. After metaphase is anaphase. Anaphase is where the microtubules separate each chromosome from its homology, so they move to opposite spindle poles. Other microtubules overlap midway between the spindle poles. They slide past each other and they push each other farther apart. As anaphase 1 ends, one set of duplicated chromosomes nears each spindle pole. Telophase 1 is next. This is where two nuclei form. Typically, the cytoplasm divides. All the chromosomes are still duplicated, and each still consists of two sister chromatids. So, mitosis 2 is next. This is the second nuclear division. Sister chromatids of each chromosome are pulled away from each other. Four haploid cells now form one diploid cell. So, prophase 1 is the same thing as in meiosis 1 of prophase 1. Same with metaphase 2 in meiosis 1. But anaphase 2 and telophase 2, in anaphase 2, one chromosome of each type is moved toward opposite spindle poles, kind of just like in meiosis 1. And in telophase 2, there are four haploid nuclei, so each with unduplicated chromosomes. So this is kind of the process of meiosis 1 and meiosis 2. When the cytoplasm divides after cytokinesis, the four haploid cells are the result. And so meiosis reduces the chromosome number, and it occurs only in sex cells. And that's about it. So when you're talking about the difference between animal and plant cell division, it's mostly the major difference is because of cytokinesis, and how it occurs. In plant cells, a new cell wall is fashioned between the new daughter cells, and this is where in an animal cell, the cell membrane constricts to pinch the parent cell into daughter cells. So this is about all I have. Thank you for listening to my project. Go Meiosis.